exciting session because there's a lot going on in all of the areas that are going to be discussed in this session. The session is gene therapy and drug development, and the panelists are all not only experts, but really actively involved in developing treatments for the types of CMT that they're working with. So we have, starting with Dr. and Professor Jun Lee, who's chair of neurology at Wayne State, and he's going to tell us on all the exciting work going on with CMT4J. And, excuse me, Dr. Lee has been involved with CMT4J since its discovery. I think it was in 2007, with the first patients that were identified with this. And he's carried on this work all the way up to really working to get ready for clinical trials. Next, we have Alessandra Bellino from Milan, the Ospedale San Raffaele. And Alessandra has been not only working to develop treatments for the CMT4B disorders, but she and her colleague Davide Perrison have done an amazing job for this very rare disease of just getting together patients from all over the world so that true natural history data can be discerned to power these trials. And then finally, we have Cleopas Cleopa from the Cyprus Institute of Neurology and Genetics. And Cleopas has really pioneered gene therapy replacement for CMT4C, but also in CMTX, and the whole approach of using adeno-associated viruses and lentiviruses to develop gene replacement for CMT models, and is really leading the field in this area. So I'm excited to hear the talks. I'm going to shut up and see if I can get off the screen, and we'll start with Professor Lee.
So the CMT4J, but in general, CMT type 4, in my opinion, is probably one of the relatively easier disease to subject to the gene therapy, because most of them tends to have what we call loss of function. That means both gene copies got problems, and their mutation end up produce very minimal or no proteins. So one could produce the gene therapy against the disease by replace the loss of the protein or replace the loss of the gene. So for the CMT4J, just briefly introduce the disease. It's caused by mutation in a gene called FIG4, F-I-G-4. And usually it's caused by genetic mutation called compound heterozygous mutation. That means mutation in both copies are different. One of the copy could be, say, missense mutation, which means only one of the amino acid got changed. And then the other copy maybe have a truncation mutation, which means at one of the codon got stopped translation, so therefore no protein gets produced. And then even the copy produced the missense mutation, but the protein is so unstable. So in that situation, that protein gets digested in the cell very quickly. So the net effect is that these disease mutation produce zero or minimal amount of the protein. So that's a loss of function. So due to that mutation, and this disease usually produce very severe demyelination, means that the insulating material called myelin wrapping around the nerve fibers, and those myelin gets degenerated, and so they cause a problem for nerve to function. And in addition to that, and there's also have severe neuronal degeneration, means the neurons is dying, and all this combination of pathology end up patient have very severe sensory loss and also very severe muscle weakness. And so those patients usually start with their clinical symptoms from the childhood, and then progressively getting worse. So over the past several years, there have been variety of strategy developed to devise the gene therapy against this disease. And obviously, the success of AAV viral vector is one of the major groundbreaking advance in the field, as you guys all know. That the success story in the spinal muscular atrophy has been inspirational. That means that they're using this viral vector, carry the missing gene of the spinal muscular atrophy into the neuron. They were able to dramatically improve the patient clinical deficits, life expectancy, as well as pathology. However, like everything in the world, that nothing is perfect, and every new method gets invented, and then later on there's always some shortcomings realized. The same thing for every viral vector. There's a significant bottleneck in the viral vector production, and usually it's very costly. And then the AAV viral vector, many of them may produce a permanent DNA alteration. So if there is an off-target effect, then you will have a potential dangerous outcome. And then over the time, and there's some realization that when the gene bring into the human cell, if they produce too many of that kind of protein, and they may also produce toxicity. And but nevertheless, and this is one of the major advances in the field. So they have been group of scientists produced the mouse model for CMT4J, and then the vector carry the gene called Fig4, also being produced and injected into the mouse model, and then the pathology and clinical deficits of the mouse are significantly improved. And that study has been recently published. There is a very important biotech company called Neurogene now have been actively involved in this gene therapy effort. They are partnership with a variety of scientists, and they try to advance this. And one of the effort they've been doing is to do the natural history study. And also through the natural history study, hopefully there's also have improvement of the outcome measurement. And then and hopefully that in the near future that gene therapy can be translated to the clinical use. CMT Association also initiate a partnership with Tesha on the CMT4A gene therapy, and that's a different disease, which I will not discuss in any detail. And I do want to spend a few minutes on this concept called gene bandage as an alternative approach, and which I feel is quite exciting. So these concepts are many derived from this publication in the New England Journal of Medicine in 2019 from a group of scientists led by Dr. Tim Yo at the Boston Children's Hospital. They were successful for treating a disease called Bateman's disease, where it causes very severe seizures, and then patient died very early in their childhood. They were able to start from the diagnosis of a child, and from November of 2016, by the time they start treating first dosage of the patient, and the total length is only about 1.5 years. So it's very remarkable how quickly they can achieve this. But what is more importantly, and I think this is something that can be utilized toward the treatment of CMT4J, some of CMT4J, or many other CMTs, is the strategy they're using is really interesting. And what they find out for this specific disease, the beta disease, is there is a mutation called splicing mutation. And obviously, there's a mutation sites which they identified. And the bottom line is through this mutation, they end up produce the abnormal splicing, which means the protein is abnormally produced. So what they did is they target on this area where they have a splicing mutation, and they produced the oligo, antisense oligo, and where they patch the area where they have mutation. So they produce several of those, and what they find is one of them. I think that this is the one that end up have the highest efficiency of correcting the mutation, and they showed the pathology is improved initially in vitro study. And then what they did is because they were able to patch this area where that splicing mutation are no longer be able to splice the exon out and then produce that mutation, so that a gene mutation gets corrected, and then pathology gets better. And then the patient, they design this oligo and inject into the patients, and then there is a dramatic clinical improvement. For example, that this is the percent time span in seizures drastically improved. And the exciting thing about this is there's no bottleneck of the production, and anti-oligo can be easily manufactured very quickly. And number two, these each injection they can last several months in terms of their effective. So you need repeat the treatment, but you don't do that like every day. So it's a long-lasting effect. The thirdly is that if there is any off-target effect, and it's temporary modification, so that if there's off-target effect, it will not produce a permanent problem. So all these are the good features of these. And many of the mutations CMT patient can be treated with this strategy, and including some of the CMT4J mutations. And the other potential caveats about this is at this point, now this bandage strategy may not be able to target on every kind of mutation, and some of the mutation still needs to be studied and figure out the better strategy to target on. So right now, this treatment only limited to certain specific mutations. So I was told by CMT Association staff and told me that I should mention a few things about another disease called HMPP, which is hereditary neuropathy with liability to pressure palsies. And as you guys all know, that this disease is very unique, in my opinion, does not belong to any classification of any CMT. It's a very, very unique disease. Clinically, they tend to produce focal sensory loss or muscle weakness when the nerve exposed to the mechanical pressure in arms or in legs, etc. But time goes on toward the later stage of the disease, patient may produce symmetric sensory and loss and muscle weakness. It will look more like other type of CMT. And the pathologically, and they usually produce a pathology called temacula, which means there is excessive myelin buildup in the specific area of the nerve. And but this kind of pathology will gradually build up during after the birth. But what we find is there is a lot of pathology which showed up far earlier than any of those myelin axon pathology, which is the myelin junction disruption. And this is diagram showed what is myelin junction looks like. And here's the axon, and there is the myelin that wrap around the axon to insulate the axon. And then each wrap still have tiny space. And in order to seal this tiny space, the nature produced those proteins called junctions. They seal this space. What we find in the very early stage, as soon as it's born, in the first week of the animal, or in human, hard to tell how early that is, but animal model at least is very, very early. And then this junction gets disrupted. So the myelin insulation is disrupted, and then show the problem for transmitting the signal of the nerve. And we also find the pathway that responsible for this junction disruption related to a increased activity of the protein called P21 activated kinase. And we were able to devise a small molecule and to block this activity. And then turns out we made this disease function better. For instance, if we don't give the medication, and these nerve function activity that goes down. But if we give the medication, we're able to completely block this dysfunction and then maintain the nerve function to propagating this nerve signal, and the pathology also gets better. We're now in the stage of we already got this patent, and we're now in the stage to seek the partnership from pharmaceutical company to try to advance and translate this to clinical use. So I will. I think I run out of time. I will end up here, and to give it to the next speaker.
Okay, thank you, John. That was an exciting talk, and I think just because of the scheduling, we don't have time for questions at this point. If there's questions, if we have time at the end, we'll move there. So we're going to have Professor Bellino go next. So.
Okay, thank you, Mike. Try to share the screen.
You okay with that?
Yeah, let me see if it's working. And is that okay? Can you see it?
Yes, now.
Okay, fantastic. Okay. So thank you very much. My name is Alessandra Bolino, and as Professor Shai said, I'm working in the San Raffaele Scientific Institute, which is an hospital in Milan, in Italy. And this is just to highlight what was and is my contribution to the CMT4 neuropathy field. So this is the classification, as also Professor Lee introduced, of the CMT4 neuropathies. So I, in the past year, contributed to identify these two genes here, MTMR2 and MTMR13, that are responsible for CMT4B1 and B2 neuropathies. And in particular, my group generated animal and cellular models to study CMT4B1, in particular among these two. And also, I spent many years in trying to characterize the role of MTMR2, which is this protein here. But then my lab also contributed to study CMT4J, and in particular, generating animal models for CMT4J. So for example, by mutating the protein of interest, that is FIC4, specifically in Schwann cells, which are the cells that produce the myelin and the PNS, as you know. And also by suggesting that MTMR2, which is this protein here involved in CMT4B1, and the FIC4, which is involved in CMT4J, are indeed functionally connected, at least in Schwann cells. So specifically, I have the honor to be supported by the CMTA this year, actually last year. And the aim of the CMTA project in which I'm involved in is to assess whether niaspan, which is an FDA-approved drug, actually, can represent an effective treatment for CMT4B1 that I presented you before, but also for other neuropathies which are characterized by hypermyelination, which is a form of myelin degeneration. So CMT4B1 is a neuropathy, is an autosomal recessive, severe demyelinating neuropathy, which has a childhood onset for most cases, and is caused by loss of function mutations in the MTMR2 that I introduced you before. Loss of function means that the protein is not produced in all cells, but only the peripheral nervous system eventually is affected in these patients. MTMR2 is a particular protein, so is an enzyme that acts on lipids as a substrate and not proteins, as usually you can have described. And this neuropathy is also caused by the presence of these myelin outfoldings in the nerve. So here you have an example. So if this is a myelinated fiber, so this is the axon and this is the myelin, the insulator of the axon, this extra loop is not normally present. So this is a form of aberrant myelination, we say, that can be even more complex than this, as in this case. And this then tends to degenerate with the progression, causing, of course, problems in the nerve, as you know. So over the years, we generated mutant mice models for this neuropathy. So several mouse models that lack MTMR2, so the protein of interest, as well as cellular models that I will not enter into details in, but just to say that all these tools allowed us to understand a bit more of MTMR2 function. And indeed, after many years here again, unfortunately, we were able to propose that MTMR2 together with MTMR13, which is the other member of the other protein mutated in the CMT4B2, what it does is that acts on these lipids that are here are in green. And these lipids in turn have the ability to increase a signaling, so a cascade of molecular events that can control myelin growth during development. So not throughout the fiber, this is a myelinated fiber, but particularly we hypothesized around the node of Ranvier, which is here, which is the portion of the axon where the action potential takes place. So when MTMR2 and R13 also are lost, what it does is this signal is overactivated, and this is creating a loss of control of membrane growth, thus causing myelin outfolding. So this aberrant myelin that I showed you before. So we designed several therapeutical strategies for CMT4B1 based on what we knew and we know now on the pathogenetic mechanism behind, and particularly using drug inhibitors for these specific pathways that I showed you before. And also, we are interested in developing strategies that are mechanism independent. So this means not dependent on the specific knowledge of what is causing molecularly the neuropathy. One is the gene therapy, so it's based on enzyme replacement, since this is an enzyme as a phosphatase, and it should be ideal to replace since it's lost in the disease. And then a drug repositioning we call strategy, so try to understand whether drugs that are already approved for other purposes, other means, can be effective also for this neuropathy. And this is a project that started many years ago again, that has been funded to generate preliminary data by Teleton Italy, and now the Italian Ministry of Health, and of course you all, CMTA. So what this project is aimed at particularly. So as I said before, to assess whether this niaspan, which is an FDA-approved drug, is effective for the treatment of CMT4B1, so the one with the myelin outfolding and MTMR2 loss. So niaspan is a commercial name of an active principle, which is niacin nicotinic acid. And what we aim to do is to perform preclinical trials, of course, using the models we generated, by administering, actually, we cannot administer niaspan as it is because these are big pills that are used for humans. So what we asked at the University of Milan, our pharmacological pharmacologists as collaborators, to develop this, I don't know if you can see here, microspheres that are like micropills that are mimicking the human drug because have a long-lasting formulation. So the drug can be released in time. And so we plan to administer these pills here, micropills here, that are called Niacot, to the mice to try to see if this drug is effective to ameliorate the neuropathy. And then, as Professor Shai also introduced, our project is aimed also at performing natural history studies on CMT4B1 and B2 patients to identify, this is much important, outcome measures to monitor efficacy of therapeutical strategies, because nothing basically is known on this neuropathy on that sense. And this will be done in collaboration with Dr. Davide Parison in Milan as well. So what we are now, so the preclinical trial has been already made. So we did 5 months of consecutive daily treatment to the mice. And now we are analyzing all this stuff here to assess efficacy. So basically, we did already the so-called target engagement. So these are measured to see whether or not the compound really reached the nerve and is there, so was well administered to mice. And this was successful, I can say to you, anticipating results. And the particularly expression study that we did in the nerve in collaboration with Professor Svaren. Then neurophysiology, that is very much important, as you know, to monitor the physiology of the nerve. So if the treatment is improving nerve function, and this was positive as well. And we have in progress other analysis that you see here, and of course, also morphological analysis to score these myelin node foldings. So this is an important slide because I would like to share with you the idea that if this treatment will be effective for CMT4B1 and B2, and we have the model to test this also on B2 neuropathy, this can be hypothetically, and we show this already with preliminary data, effective for HMPP that was introduced by Professor Julie, that is characterized by the presence of tomacula, and also other neuropathies of the CMT4 group that are characterized by myelin node foldings, as you can see here, for example, CMT4H or CMT4C in some cases, and CMT4F. So with this, thank you for your attention, and I'm happy to have questions if there is time. So now I try to stop sharing. Okay.
So we thank Professor Bellino for a lovely talk, and we're still running a little bit late, so I think we'll go ahead to the last presentation, and if there's time at the end, we'll take questions. So I think we have hopefully Professor Cleopa's talk ready to go here. Thanks again, Alessandra, that was great.
Thank you.
Good afternoon. My name is Cleopas Cleopa. I'm a neurologist and professor at the Cyprus Institute of Neurology and Genetics. Thank you for joining us in this session about CMT4C. And I would like to thank the CMT Association for giving me the opportunity to provide this update on our CMT4C gene therapy program. CMT4C inherited neuropathy is actually the most frequent among the overall rare recessive demyelinating neuropathies. This is a disease that starts in childhood with a range between 1 and 14 years of age for the disease presentation. And the symptoms are well known to all of you. There is weakness and progressive atrophy of the muscles in distal limbs, loss of sensation, When we measure the nerve conduction studies, the conduction velocities are very slow, and it is not uncommon for patients to develop spine deformities, scoliosis, deformities of the feet, as you can see in this picture here, or even involvement of cranial nerves, presenting as atrophy of the tongue, of the facial muscles, hearing impairment, dysarthria, and even respiratory problems. What causes CMT4C? As with other types of demyelinating CMT neuropathies, the causative gene is expressed by the cells that actually are responsible for forming the myelin sheath around the peripheral nerves, the Schwann cells, and the responsible gene is the SH3-TC2 gene encoding the protein that has the same name, and the disease is transmitted in an autosomal recessive manner. The SH3-TC2 protein is an important molecule that regulates the vesicle recycling system in the Schwann cell. We call it endocytic recycling compartment. And this system is very important for the proper growth of Schwann cells, for the proper formation of the myelin sheath, and the maintenance of the myelin sheath. Therefore, mutations in SH3-TC2 impair the function of the protein at these vesicles, impair the communication between the Schwann cell and the axon, and cause abnormal myelination and progressive destruction of the myelin sheath, and subsequently, of course, of the axon itself, leading to the manifestations of the disease. So the strategy for treating CMT4C is to replace the SH3-TC2 gene in Schwann cells, because this is where the function of the protein is needed. And therefore, we designed a treatment approach to target the delivery of the healthy human SH3-TC2 gene to Schwann cells. Excuse me, in order to achieve that, we use a cell-specific promoter element. This is a DNA sequence that restricts the expression of the gene only in this cell type, the Schwann cell. The gene has to be delivered by a viral vector that is used as a vehicle to transport the DNA into the cell. And the second important challenge for the gene therapy here is to get this viral vector into as many Schwann cells as possible along the body. And so we did a lot of work to optimize the delivery approach using either intravenous or intrathecal or direct injection into the nerve of the vector. We finally came to the conclusion that the injection in the lumbar intrathecal space is the most effective to target the nerve roots and the peripheral nerves using the minimal amounts of the viral vector. So this is the approach we're currently using in the CMT4C gene therapy program. This diagram here shows the design of the DNA we package into the viral vector, consisting of the coding sequence of the human SH3-TC2 gene. And this is now under the control of a cell-specific MPZ or myelin protein 0 promoter that drives the production of this protein specifically in Schwann cells. When we package this gene into the adeno-associated viral vector and deliver it to mice that are deficient for SH3-TC2 protein, we can then see that the virus leads to the production of the healthy protein in Schwann cells. This is shown here in this red fluorescent signal in lumbar roots. So these are the roots near the spine, and also the peripheral sciatic nerve and in the individual fibers. So we demonstrate re-establishment of the expression of this protein in the cells of interest. Using this approach and the adeno-associated viral vector 9, we then proceeded in a project funded by CMT Association to treat the model of the disease. The model of the disease is a mouse that is deficient for SH3 TC2 protein that develops a very similar pathology as the one seen in patients. And we treated these mice both early as well as late during the disease course in order to demonstrate that we can achieve a therapeutic benefit also after the onset of the nerve pathology. We have almost completed the analysis of these treatment trials. We were able to show, first of all, by several behavioral studies, that there is a significant improvement in motor function in mice that were treated with a therapeutic vector. This is shown with the rotating rod test, where treated mice were able to last longer on the rotating rod compared to the fake or mock treated mouse groups. And also treated animals developed a higher, a stronger force in the hind limbs measured with the grip strength test. You could also see the improvement when you just look at the mouse and hold the mouse by the tail. This is a mouse that has not been treated, that has this disease because it is deficient for SH3 TC2. You can see the abnormal clenching of the toes and the hind limbs when the mouse is held in this position. In a mouse that received the gene therapy treatment, this phenotype has improved. The animal is now able to stretch the hind limbs, open them, and open the pose, indicating improvement of the strength and the function in the limbs. The improvement in nerve function was also demonstrated through measuring motor conduction velocities that were significantly increased in treated model of CMT4C compared to the untreated animals. We also looked at the morphology, both in lumbar spinal roots, but also in the peripheral nerve. In the root, we see that there is a significant improvement in myelination, in the formation of myelin. There are fewer fibers in treated mice that are completely demyelinated, and this can be measured. And you can see that treated mice have lower percentage of abnormal fibers compared to the mock treated, an example of which is shown here. We see the same benefit also in a peripheral nerve, the femoral nerve that supplies the thigh muscle. We see significant improvement in myelination after treatment compared to no treatment. We also looked at a particular pathological feature of CMT4C neuropathy, which is the elongation of the nodes. And this is a picture from a patient nerve biopsy showing this elongated node. The same abnormality is found in the mouse model of the disease, and we can measure this after immunostaining. You see here an untreated knockout model with the elongated node, and in a treated mouse there is significant shortening of the node similar to the healthy mouse. So there is also correction of this so-called nodal phenotype. So we have now very encouraging preliminary results of treating the CMT4C model with our gene therapy approach. What is the path forward? This is what we have so far. We have preliminary proof of concept data using the AAV9 vector, which is a clinically relevant vector because it's already used in clinical trials for other gene therapy programs. We signed earlier this year a licensing agreement with a gene therapy company in order to support further experiments and the development of the program for clinical translation. So this company now is funding further experiments, including a dose response study. So it's a dose escalation study comparing 3 different doses of the vector to see which one is the most effective and safer. We also do some toxicity analysis when we inject the vector. And this work, hopefully within next year, will provide a complete set of data to provide the proof of principle for moving forward with the gene therapy for 4C. At the same time, the company is planning to start a natural history study for CMT4C to enroll patients. This is also very important. And then after completing the final steps, including evaluating toxicity and biodistribution in larger animals, then this will lead to the IND, investigational drug filing, and hopefully in the not so distant future, the initiation of a clinical trial for CMT4C. What is important is that we have to also have clinical trial readiness while we complete our preclinical experiments. And currently, we only have cross-sectional data available indicating the severity of the disease. So it is important to characterize more sensitive clinical outcome measures and to obtain longitudinal data of disease progression, which is something that will be done with a natural history study. And this longitudinal study will also include novel clinical evaluation tools like CMT functional outcome measures, health index, and other measures of disease severity. Future clinical studies will also include novel biomarkers that are more sensitive in measuring disease progression and response to treatment. One of them is the amount of fat replacement in the muscles that become weak. And this can be done with a magnetic resonance imaging. It has already been established in other CMT neuropathies. And we will also see the introduction of blood biomarkers that reflect damage in the nerves. One of them is the neurofilament light protein, which can be detected elevated in blood of CMT patients. And we already showed in previous work that it is elevated also in the model of CMT4C and improves after treatment. So it is a treatment responsive biomarker. And further novel blood biomarkers are being studied to increase the tools that we have available in order to optimize the design of future clinical trials and to be able to demonstrate the therapeutic benefit of the gene therapy. I would like to thank you for your attention and for joining this session. And of course, I would like to thank the people in my group that provided this data and the Charcot-Marie-Tooth Association for supporting this study. Thank you very much.
So thanks, everybody. Jonah, are you with us? So I don't think we have Cleopas. I think that, hi, Amy, can you hear me?
I can, thank you.
Yes, you can. Do we have time for any questions?
We have time for one question. If we have a question from the community, I don't see any questions right now in the Q&A though.
Well, I have just a quick question for Professors Bellino and also for Professor Lee, that these are exciting times. These are rational therapies, and people with these diseases obviously are excited to get clinical trials underway. Do you have a sense, starting with Professor Bolino, of the time frame where we could actually see clinical trials, initially with the CMT4Bs, and also Professor Lee with CMT4J afterwards?
So the first, as I said, so we are at the level of proof of concept still at the preclinical level. So this will take at least another, I think, a year, maybe. In the meantime, we are trying to, do natural history study to set outcome measures. Otherwise, the clinical trials cannot be performed and the efficacy cannot be tested in humans. So if I can guess, would be another couple of years, maybe.
Okay, thank you. And Professor Lee? I think you might be muted.
Every time I try to predict something, I end up always wrong. So, I think that the situation with CMT4J, due to the partnership with Neurogen, it has been advanced faster than initially, at least initially faster than I thought. Everything has been progressed well. However, there have been the variety of other issues. I think the most challenging one is the realism of the disease, and then recruiting the patients is one of the biggest challenge, I think, because it's so rare. The other thing is that we do not have any biosafety data for that specific vector that we're using. So that's also another potential roadblock. And FDA is also very keen, and they really want to have a better outcome measurements, and that's also another potential roadblock. So if my guessing, I will say probably at least another 2 years, probably even longer.
Okay.
Thank you.
And I don't want to give the impression that this is just, everything's always 2 or 3 years down the road. I think these have shown, all these approaches have shown real progress, and these are legitimate questions that have to be answered, but they're being answered. So again, I think we're on the verge of getting really solid clinical trials in these diseases. And I thank all 3 investigators for all the fantastic work they've done over the years. And even though I'm involved with the CMTA, I really want to thank the CMTA for their support of these.
Thank you, Dr. Shai.
Yes, indeed. Thank you very much.
Thank you.
Thank you, everyone. And if those that are still in this session want to head over to the biotech panel, you'll get to hear a little bit more directly from some of our biotech partners about the work they're doing to develop therapies for CMT patients. Thank you again.
See you.
Okay, thanks, Amy.